Bennett International Transport is a premier provider of logistics, transportation, and cold chain solutions to government contractors and commercial businesses for over 46 years. Bennett combines every aspect of global transportation to provide innovative and cost-efficient solutions to deliver your products safely to the market. When you're transporting chemicals, pharmaceuticals, plasma, or other cold chain commodities, it needs to be perfect or it's wasted. As one of the premier cold chain solutions, transportation, and logistics provider to the protein and biotherapeutic industries, it means you can trust us with your most delicate and important jobs. We have the in-house expertise to handle time-sensitive and perishable shipments. We offer seamless service for both deep frozen and perishable moves, utilizing specialized equipment from our ocean and air freight partners. Our main focus is to ensure that your cargo is perfectly protected at every step in the process and kept at its needed temperature. Our project managers know the regulations and packing requirements, and our commitment to transparency and visibility means you'll always know where your freight is and who to contact with any questions. Our primary focus is providing ocean and air freight service globally. We have a specialized team and worldwide network of partners to ensure proper monitoring at all times, offering re-icing services globally to temperature control, no matter what the circumstances, for all your cold chain logistics needs. Let Bennett serve your perishable needs. Contact us today to learn more. www.bit-international.com or call 631-560-0389. With a proven track record and unmatched expertise in logistics management, Bennett International Transport offers comprehensive global transportation services to meet your needs. We are proud to continue our support of attending PPTA and IPPC forums yearly as an exhibitor and can't wait to see you next year.
Good morning, good afternoon, ladies and gentlemen, depending where you are in the world. My name is Domenica Mistela. I'm PPTA's Senior Director for Regulatory Policy in Europe. It is my pleasure to introduce you to the next session entitled Experts on Coronavirus, the Virus, the Disease, Prevention and Treatment. Certainly we have learned a lot about the virus and the disease since the beginning of this year up until now. We, in the meantime, have reached a pretty grim milestone of globally over 35 million persons infected and over 1 million of lives lost. We have, however, also made significant advances in how to test, diagnose, and even treat people. Nevertheless, there are many gaps in the knowledge as we continue to learn about the disease and the virus. I'm very proud to now introduce three esteemed experts who each in their own capacity are helping to build this knowledge. I would like you to reserve your questions for after all the talks. We will hold a question and answer session at the end of the talks. So I would like to introduce the first speaker. It is my great pleasure to welcome the esteemed expert, Dr. Thomas Krail. Dr. Krail is Associate Professor of Virology at the University of Vienna and Vice President for Global Pathogen Safety at Takeda. Dr. Krail will present to us some crucial details and learnings on SARS-CoV-2, the virus responsible for the COVID-19 pandemic. In his role, he has stood at the forefront of research of this virus in conjunction with safety of plasma protein therapies as early as January, February this year, and as well as recently his work on the development of therapies against the virus. Beyond his very well-known work at Takeda, he's also the chairman of PPTA's Pathogen Safety Steering Committee. He's also a member of the steering committee of the Consortium on Adventitious Agent Contamination and Biomanufacturing, coordinated by the MIT. Dr. Krail has a PhD in biochemistry from the University of Vienna and spent several years as a postdoc at the same institute focusing on antiviral immunology and specifically the role of antiviral antibodies. He then joined Takeda, previously known as Immuno, Baxter, Baxalta Shire, and has been ever since working in the pathogen safety of biopharmaceuticals, as well as research on virus antibodies and vaccines. is certainly very well regarded and very well published when it comes to pathogen safety of plasma protein therapies and biologics. And most recently, his lab has been very prolific when it comes to publications on SARS-CoV-2. And when we were all allowed to travel globally, Dr. Kral has been a frequent international lecturer. Thank you very much, Dr. Kral. The podium, the screen is yours.
So dear Dominika, very kind words indeed. I'm not sure I deserve any of them. A warm welcome also from my side, while unfortunately only virtually. What I would like to discuss with you today is what we know about the SARS-CoV-2 virus, the virus that causes COVID-19 around the world, what is known already, and maybe a few aspects that we have not yet found out. It is hard to believe that there are still things that we don't know about the virus, because ever since January this year, 35,000 publications have become public around this new virus, which is just, I mean, it's too much to even catch up reading. Anyway, let's start in the beginning. In the week of January 6th this year, we had our weekly review of any indications of emerging viruses. This is something that we do every week. And in that week, we had a report from WHO issued just the day before about pneumonia of an unknown cause in China. This goes back to where in December 31st, the WHO China office was informed that there were a few people suffering from an unknown pneumonia with no etiology identified at that stage in the Wuhan province of China. The stage at that point was that 44 people had pneumonia. Some of them were severely ill, some of them in stable condition. And it was all linked to one of these notorious wet markets in China where live animals are sold for food, actually. And the market had been closed already, but that was all the information at that stage. A few weeks later, then, we had the first what we call Quality Council, where I gave a pathogen safety update. And it again related back to that report out of China to the WHO. But 3 weeks after that first consideration, we had already confirmed almost 8,000 cases in China and a total of 170 people dead. I think it is important to stress that at that stage, everybody thought this was something that was purely China because other locations were plus minus not affected. At that stage also, we did a review of plasma product safety margins, and as at that stage, the coronavirus, the causative agent, had already been identified, and we knew quite a bit about the physicochemical composition of these viruses. A first assessment ended up saying that plasma product safety was uncompromised. I'll come back to that in just a few minutes. So time has gone on, and this is where we stand now. As Dominika has mentioned, this virus has caused a pandemic. So it is now a virus that really affects all people, pandemic. That's the source of the word. So now we have 35 million around cases in the world. We have really seen an unfortunate 1 million deaths. And what we see also is that China, where the virus originally came from, is actually not the epicenter at this stage. Actually, China had less than 100,000 cases with still an unfortunate 5,000 deaths. But where the pandemic right now rages is more in India, Brazil, so countries with a somewhat less developed healthcare setting maybe. What is more surprising yet is that the lead is taken by the United States of America, with more than 7 million cases there, and again, very unfortunately, more than 200,000 people dead. I think the most unsettling part of that graph from the Johns Hopkins University is this part here, in that there is still no end in sight. Case numbers are still going, and it begs the question almost whether with the onset of the cold season in the Northern Hemisphere, we're not going to potentially even see an acceleration of cases. So truly unsettling. While I am a virologist, I will make mention of the fact that beyond the virological concerns, there is these economic concerns. I think the virus has the potential to result in a lot of second thoughts about globalization altogether. While globalization sounded like a great concept in that it allows you to increase buying power by sourcing your materials from elsewhere where you can get it cheaper, I think it has become clear that that same globalization has resulted in a lack of resilience in the supply chains, for example, of medicines. If you depend on people in India or China to produce the raw materials that you then can finish your products from, then that is, in a situation like the one we're facing right now, probably not the most smart choice. Just to illustrate, in a recent conversation with the CEO of Novartis here in Austria, he mentioned that if you expect a pack of antibiotic to be cheaper than a pack of chewing gum, then you cannot expect that we will produce that in Austria. It has to be produced elsewhere. And that's why the pharma supply chains, I think, have illustrated for us that this globalization comes at a price. Now, back to the virus. I think the most noteworthy element around that virus is that it should not have been a surprise at all. We have had the first SARS virus some 17 years back. At that stage, it was not known that these viruses could occur in bats. Ever since, we have identified 30 plus different coronaviruses in bats alone. So it was quite clear that there was an abundant supply of these viruses out there, and it was quite clear that they had the ability to be zoonotic, so jumping over from an animal to a human host. We have seen that occur at least 8 times by now, and in some instances, these viruses cause really bad human diseases. SARS, MERS in circulation in Saudi Arabia, and now SARS-CoV-2. So I think it should have been quite clear that there is a concern out there, only we were not prepared for it at all. And I think right now we are facing the consequences of this. Maybe a learning out of there that next time around we should be more prepared, but that's maybe a different thing. So more details specifically on the virus, the SARS coronavirus 2. One of the problems with that virus is that it's a biocontainment level 3 virus, which has limited the places that can work with this virus to actually rather few labs. And that's why it has been difficult to answer some of the most important, the most pressing research questions around the virus. It is a beta coronavirus, and yes, there is alpha coronaviruses as well. And it's a beta coronavirus like the OC43 and the HKU1 viruses that are in circulation in humans as seasonal flu-like illness viruses. But for an oddity, it uses a cellular receptor that is called ACA2 that is used by other coronaviruses. So OC43 uses a different one, but the NL63 uses this receptor, the same receptor. The reason for mentioning is it's a question what the closest relative to the new SARS virus is going to be. And it's going to be interesting for us, I think, to figure out the level of serological cross-reactivities. And I'll come back to that in just a minute. On the modes of transmission, we have learned, meanwhile, that aerosols and fomites are responsible for the transmissions. So the protective measures are clear from that, in that for aerosols, you want to wear masks. In my personal opinion, you do not want to wear face shields because there is good studies out there that have illustrated how poorly protected these are. And then the second thing is washing hands. I think both are really important. And it's intriguing to realize that in the Southern Hemisphere, flu has not been quite as impactful in this gone season. And in large part, I would argue that is just because of the better hygiene that the SARS coronavirus has driven. So maybe washing hands is something we should have adopted also earlier. And then finally, to the virus itself. It is lipid enveloped and is around 120 nanometers or even larger. So it is very susceptible to many treatments that we use in our manufacturing processes for plasma derivatives. And it's large, so it's relatively easily removed. And that was, both of those were actually very reassuring aspects to realize. And I will say that PPTA and the Pathogen Safety Steering Committee have done their job. Again, very early in January, we first heard about that virus. And by January 29, the Pathogen Safety Steering Committee had pulled on the website of PPTA a statement summarizing that after review of all the data that is available, it is clear that this virus is not a concern for the safety of plasma protein therapies. So how did we come to that conclusion? First, for respiratory viruses, it is not known that infectivity can occur in the blood and therefore also plasma. And then secondly, the inactivation would work fine with a lipid envelope virus. And also the removal should work fine for a very large virus. So data that are specifically around the SARS coronavirus 2 are not available, but there is other coronaviruses that have been studied. And again, very reassuring evidence, exactly they behave like expected, and therefore the safety margins of plasma derivatives not compromised. Now, plasma derivatives is certainly what this audience is most concerned with. I will say, though, that the concern doesn't stop at plasma products. Equally, for recombinant biologics, questions were raised. This is a recent guidance from FDA where they have asked, are production cell lines may be infectable with that new virus? And would the adventitious agent testing in place detect such a contamination if it were to occur? Now, this is a study we've done here in the house. Taking a look at the production cell lines that we have in use, we could confirm that they would not be infectable by the new SARS coronavirus. Taking a look at the cell panels that we're using for adventitious agent testing, it's quite clear that Vero, that is part of all of these panels, very easily detects the virus by a cytopathic effect as well as by heme adsorption. So quite clearly, these were not concerns, and the virus inactivation and removal I have just earlier mentioned. So what even happens when a person is infected with the new virus? This here is, I think, a quite intriguing case where an Austrian physician was collecting CME credits, doing that in the Austrian Alps in a nice resort. But that resort was not so large, and that's why this gathering resulted in one of the first clusters here in Austria. The fortunate part of it for us was that he also, during quarantine, had a wife, and she was a medical doctor too. So he volunteered to get his blood drawn every few days. And so we could exactly reconstruct the course of disease. So it started with a bit faintness. Then he was diagnosed. He developed a dry cough. And around day 10 or so, that was the first sample where we could detect neutralizing antibodies to the virus coming up. So the neutralizing antibody titer rose very quickly up. He had a few other symptoms like pressure on the lungs, diarrhea, and argusia. Don't mind if you don't know the word. I didn't know it either. It's a loss of taste and sensory sensation, so you don't have any feeling for appetite anymore. And then here the person was symptom-free, and the antibody levels started to decline somewhat. It's not a big decline, and it's something that you would expect actually in every infection after the acute phase is over, some level of decline of the antibodies. Now, the antibodies do have a role in that for this infection, they were able to clear the virus. And I think there is a therapeutic aspect to that as well. You're going to hear a lot more about this in this meeting. I'm just going to briefly touch on it. So if a person is infected and then gets ill, after a while, the people get hopefully healthy again, but now they carry antibodies. And if these people can then be motivated to donate plasma, then that's what we call convalescent plasma. And that in itself can be used as a treatment. If that plasma now is fractionated into immune globulins, then what the result is a hyperimmune globulin. And again, this is something under consideration for treatment. So in the early days, the only thing that was available was convalescent plasma. But there was a need to confirm that for these convalescent plasma units, you had potent neutralizing antibodies in there. And that's why we had a collaboration with the Austrian Red Cross, where we tested the distribution of these neutralizing antibody titers. They're at around 230, which is somewhat higher than plasma for fractionation. And the reason is there that the Red Cross prefers male donors of somewhat advanced age because of compatibility issues, and those tend to have a higher antibody titer. Now, speaking about antibody titer, people were thinking, if there is so many coronaviruses in circulation already in the plasma donor community, could there not be cross protection? And that was studied as well. Here you can see this is immune globulins produced from plasma collected before the pandemic, and it has 0 neutralizing antibody titers against the SARS coronavirus 2. The same IVIG lots tested with 229E, which is one of these seasonal coronaviruses, has very potent neutralization. In other words, there is no cross-neutralization between these viruses. And similar investigations with other viruses are going on. This one has actually been confirmed by Florian and Kramer's group at Mount Sinai in a very nice Nature paper, and similar work out of Japan is going to be published soon. With that, I would already like to conclude. As Churchill has said, never let a good crisis go to waste. So there is learnings that have come out of this. I think for the Pathogen Safety Steering Committee, we were able to do the right thing in that we jumped to action quickly when that concern arose, and therefore were able to confirm that the products produced by PPTA member companies anyway are safe. Complacency in this industry, never. We know our past and we shall not forget. And now the fortunate aspect of it is maybe that plasma-based treatment options will even help get over this pandemic. Thank you very much indeed.
It is my pleasure to welcome as the next speaker, Dr. Louis Katz, who will provide an overview over the many clinical aspects of what a COVID-19 infection can do to a human, how we can test for it, and how we can potentially treat it. Dr. Katz is the acting chief medical director of the Mississippi Valley Regional Blood Center, Central Illinois Community Blood Center, Community Blood Services of Illinois. So it provides blood and blood products services to over 110 hospitals in that region. Dr. Katz is a board certified internist and infectious disease specialist who trained at the University of Iowa. He's currently adjunct clinical professor of internal medicine at the Roy and Lucille Carver College of Medicine. Dr. Katz is very well known to the transfusion community. He's the chief medical officer at America's Blood Centers in Washington and ABC past president, former board member, and past chair of Scientific, Medical, and Technical Committee of ABC. Certainly his expertise in transfusion medicines and especially in transfusion-transmitted infections spans several decades and also several pages of peer-reviewed publications and numerous book chapters. Dr. Katz served multiple terms as the chair of the AABB Transfusion-Transmitted Disease Committee on various other AABB committees, and also as a member and chair of the FDA's Blood Products Advisory Committee, and is currently member of the HHS Advisory Committee on Blood and Tissue Safety and Availability. Dr. Katz, the screen is yours. Thank you very much.
Thanks to all my friends at PPTA for the invitation to do this. I'm going to talk about clinical aspects of COVID and some of the treatment options. These are my disclosures, nothing that should be a conflict of interest. Transmission, you've heard a little bit about, primarily droplet and contact. Aerosols are important. We don't know how much. The role of the environment and fomites, unclear, but probably minor. Coughing, sneezing, singing, talking, breathing, the activities of daily living are associated with transmission. The amount of airborne as opposed to droplet transmission is really poorly characterized. It's important, but how important we're not sure yet. And of course, the relative role of airborne as opposed to droplet transmission has a large impact on the mitigation interventions that we consider most important. The virus is largely gone from respiratory samples by about day 9 or 10 after the onset of symptoms. People are no longer infectious. That correlates with the rise in neutralizing antibodies that Dr. Krause showed you. Vertical transmission, that is mother to infant, appears to be quite uncommon, although isolated case reports are appearing in the literature. This is an important issue, pre and asymptomatic transmission. Pre-symptomatic meaning transmission after infection before somebody is ill. True asymptomatic transmission would be transmission from those patients who never develop symptoms. Pre-symptomatic transmission appears to occur in about 17% of cases based on a lot of data from episodes like the Diamond Princess cruise ship and other venues. Asymptomatic infections are probably a little less likely to transmit than symptomatic, less coughing, for example, with a relative risk of about 60%. I need more data to more clearly define this, particularly in subpopulations like children, who appear to be infected at rates similar to adults, but perhaps transmit a little bit less. This is my county, where I'm the health department medical director. It's our epidemic curve since March, and you can see what is beginning to look like cycles of transmission, each cycle being associated with the premature relaxation of mandated non-pharmaceutical interventions and holidays, reopening of schools and whatnot. So we hope, but appear to be in a situation where we're going to cycle through peaks and valleys in the epidemic until we have a vaccine and can begin to approach some sort of herd immunity over time. This is the age in my county, and it's very similar to national reports. Morbidity clusters in older age groups, in addition to those with comorbidities, but we're finding, particularly over the last couple of months, that infection clusters in a younger age group who tend to remain well, who tend to adhere less than optimally to non-pharmaceutical interventions like masking, social distancing, staying out of bars, keeping their voices down when they're in bars, and other behavioral things. And it's this large young adult and middle-aged cohort that appears to be driving the epidemic in my county and in the United States generally. Who gets ill? You can see that the likelihood of hospitalization goes up rapidly with age. This has been recognized since very early in the epidemic in China. And on the right, you can see the disproportionate impact of COVID on racial minorities in the United States. This impact on racial minorities appears not to be a biologic phenomenon, but a demographic and economic set of confounders where crowding in the household, insurance status, access to care, prevalence of comorbidities and whatnot, magnify the clinical impact of the epidemic in minorities in the United States. Clinical diagnosis works very well, in fact, for influenza, not so well with COVID-19. And the reason for this is almost all influenza transmission occurs with and after the onset of symptoms. So those likely to spread are easier to identify. With pre-symptomatic and asymptomatic transmission of SARS-CoV-2, syndromic surveillance becomes problematic. So we depend on contact tracing and widespread testing to identify those who are infected for isolation and those who may have had contact for quarantine. Nucleic acid testing is the test of choice for reasons of sensitivity and specificity. Turnaround time and complexity and expense are a problem. So depending on the venue in your community where you're testing and doing surveillance, the choice of tests may differ and the specimens may differ. So for example, we're trying to deploy rapid antigen testing in our schools. I think in the upper right here, you see a kind of paradigmatic clinical progression from asymptomatic infection to upper respiratory infection, to viral pneumonia, followed by respiratory failure, cytokine storm, thromboembolism, and the other serious complications being seen during the illness. The epidemiologic parameters I have shown on the bottom of the slide are evolving. Probably most important is the infection fatality rate. That is the proportion of infections, whether recognized as cases or not, who ultimately die. That's about 0.6%. It's an evolving number. That is 6 or 7 times the rate for influenza A for context. I'm an ID doc. We always think about STDs when we think about anything. When I was in training, we were admonished about Sir William Osler's aphorism, "He who knows syphilis knows medicine." You might make that case for COVID-19 here. I'm not going to go through everything on this slide, but you can see the very wide variety of organ systems and phenomenon being described in patients with COVID-19. And I most particularly highlight thromboembolism and cardiac diseases, which are emerging as major concerns in the hospital and for the long-term recovery of COVID patients. Lots of hematologic and immune markers that are being used. The importance of understanding these is as we move forward with more clinical evidence and are able to use some of these in vitro assays to predict who's going to do poorly and intervene more aggressively. Presenting symptoms you can see here, over on the left, is primarily fever, respiratory symptoms, and a whole wide variety of things moving to the right. The insert is from my county and demonstrates numbers that really haven't changed since the original Chinese descriptions. About 80% of individuals symptomatic, 14 to 18% asymptomatic, and a large majority able to take advantage of home care, with a much smaller number hospitalized and at risk for severe disease. Testing and who should be testing has become the subject of political discussions in addition to epidemiologic and medical in the United States. But essentially anybody with symptoms should be tested. This allows identification of contacts, isolation of infections, and quarantine of contacts. These symptoms are not the entire list, but they're most of them, and the kinds of fluids where PCR is most effective are listed here. Please note that in the upper airway, a lot of work being done with both nasopharyngeal, nasal, oral pharyngeal, and salivary samples. And the impetus to studying all those different samples is the complexity of getting a good nasopharyngeal swab in non-healthcare venues. This is one severity spectrum. This is from the NIH. WHO has an ordinal scale as well. And essentially, this moves from a positive test with no symptoms through the various potential clinical stages, evidence of hypoxia, to respiratory failure, shock syndrome, and multi-organ dysfunction, and allows us to stratify in clinical and epidemiologic studies the patients that we're looking at. This is the emerging framework of the phases of infection. I think this is very important for people to understand as they listen to the media. Early infection, when virus replication is occurring, respiratory or pulmonary stage dominated by viral pneumonia, and then a hyperinflammatory stage that is characterized by inflammatory and immune dysregulation, all this playing out on average over a period of 3 to 4 weeks. Obviously, antiviral therapy is most likely to be effective early when there's infectious virus present, anti-inflammatory, and immune modulating therapy later on. Throughout the course of illness, best supportive care, prevention of thromboembolic phenomenon and whatnot are critical. Mild COVID is generally managed at home in an ambulatory setting with a close generally telemedicine follow-up for symptomatic patients, especially those at risk for severe disease, the elderly, and those with comorbidity, since rapid progression is possible. Not a lot of laboratory testing required beyond the diagnostic assay, and a little supportive therapy, particularly anticoagulant, antiplatelet therapy, being used at this point in ambulatory patients. More severe infection, often in the hospital, the initial evaluation is fairly straightforward with routine tests, plus those inflammatory markers that I referred to early. Management is close monitoring with supportive care. In the second and third weeks, empiric antibiotics. If bacterial superinfection is strongly suspected, virtually all patients should get thromboprophylaxis with a heparin or heparin derivative and appropriate infection control and prevention measures to prevent spread. More severe infection is dominated by supportive care, appropriate approaches to mechanical ventilation, minimizing sedation, conservative fluid, prone positioning for hypoxemic patients, and ECMO when a mechanical ventilator is not enough. Thromboprophylaxis, again, the role of therapeutic anticoagulation is understudied. I just a week or so ago looked at the WHO database, clinicaltrials.gov, and as you can see, there are literally thousands of interventional trials going on. Investigational antivirals, remdesivir, modest activity, favipiravir, some activity, not available in the US. Convalescent plasma, modest activity, we haven't identified yet most appropriate groups to receive it. Hyperimmune globulin has been mentioned. A lot of publicity recently about monoclonal antibodies, both from Lilly and Regeneron. Then there's a long list of repurposed drugs, drugs approved for other indications being advocated in this setting, corticosteroids, dexamethasone in particular, the substantial mortality benefit in the severely ill, and most of the others really being investigated, but absent convincing clinical evidence. Children, what do we know? This is very important because for most respiratory illnesses, children are an important reservoir. Their status as a reservoir for transmission for COVID-19 is not clear yet, and so studying them is incredibly important. Of course, the pandemic has had a disproportionate impact on kids, loss of educational quality and socialization, family stressors, nutrition issues, particularly in the US, where a lot of poor kids get a lot of their calories at school. We see delays in immunization and a variety of other accidents. Best practices that we're evaluating in schools are cohorting, social distancing, masking, improving ventilation in old buildings, and screening, which we're trying to implement in the school systems in my county now against really substantial operational barriers. So I think pay a lot of attention to what you're hearing about pediatric COVID, because it's going to be incredibly important in how we try to stem the epidemic and get kids back to where they belong in the fall. Then there's long COVID, or what editorialist at Nature called the lasting misery of coronavirus long haulers. Persistent respiratory signs and symptoms, possibly cardiomyopathy, autoimmunity, chronic kidney injury, chronic fatigue syndrome like complex, PTSD, depression, and other psychosocial manifestations. On the right is a comparison of the acute and post-COVID phases in 143 Italian patients, and you can see that it's dominated by fatigue, dyspnea, joint pain, and other symptoms that can persist over long periods. In this study, this was a 2-month follow-up. This is a story being written as we talk. As an infectious disease clinician, I have seen this phenomenon in many infections. So I'm not sure if COVID is materially different than other serious acute respiratory infections. But talk to me here, what I say about many of the questions that I get asked about clinical manifestations of COVID. So that's all I wanted to say. Come and see me in Iowa on my farm sometime. You'll be welcomed. Hopefully we'll be able to travel again when we start foaling again in the spring.
Good morning, good afternoon. It is my pleasure to welcome the last speaker of this session, Dr. Marian Gruber from the US FDA, who will give us some insights on vaccine development as well as FDA's vaccine approval process for COVID-19 vaccines. Dr. Gruber is the director of the Office for Vaccine Research and Review at the US FDA's Center for Biologics Evaluation and Research. She oversees the planning, development, and administration of the office's national and international programs, which involves day-to-day management of over 250 staff engaged in the research, development, and testing of vaccines, as well as related activities for vaccines and other biologics. Dr. Gruber holds a PhD in microbiology from the Christian Albrecht University of Kiel, Germany, and a BSc in microbiology from the University of Ulm, also in Germany. To this table, or to this screen actually, she has over 25 years of professional experience and training in the research and regulation of vaccines and other biological products, including positions held in scientific research, regulatory affairs, and biological products policy. So now, Dr. Gruber, thank you very much. Please go ahead.
Well, good day. My name is Marion Gruber. I'm the director of the Office of Vaccine Research and Review at FDA, Center for Biologics. And it is a pleasure to be here today to share with you the regulatory perspective in the development and licensure of COVID-19 vaccines. So what I would like to do over the next couple of minutes is talk a little bit about the FDA response to COVID-19, our international collaborations. Then we'll talk a little bit about vaccine development strategies in general and focus on some unique features in COVID-19 vaccine development. And finally, give you a very high-level overview about the guidance for industry document entitled Development and Licensure of Vaccines to Prevent COVID-19 that we published in June of 2020. So in terms of international collaboration, we of course do engage in a lot of collaborative efforts with our national partners. But in addition, we do engage in information sharing about investigational COVID-19 vaccine constructs with other national regulatory authorities. We did lead and organize a global regulators meeting to reach global regulatory alignment as feasible for data requirements supporting COVID-19 vaccine development and licensure. And we also do participate in the ACT Accelerator, that stands for Access to COVID-19 Tools, which is a global collaboration to accelerate the development, production, and equitable access to COVID-19 treatments and vaccines. And COVAX is the vaccine pillar of the ACT Accelerator. And so we discussed the development for COVID-19 vaccines with international partners using this platform. And last but not least, we are engaged in participating in several WHO working groups, such as the Clinical Trials Working Group, the Assay Development Working Group, and the Animal Model Development Group to provide scientific and regulatory advice to facilitate COVID-19 vaccine development. There are many vaccine manufacturers who have been approaching the FDA over the last couple of months with interest in developing preventive vaccines against COVID-19. And these SARS-CoV-2 vaccine constructs are based on different platforms and technologies such as DNA and RNA vaccines, subunit vaccines, inactivated or live attenuated vaccines, as well as non-replicating and replicating viral vector vaccines. Most of these vaccines target either part or all of the spike protein of the SARS-CoV-2 virus, which is expressed on the surface of the virus. And it is the spike protein by which this virus binds to the human cell surface receptor ACE2. When we talk about vaccine development strategies and data required to support licensure, there are 6 principles that we have to think about. First of all, the development and refinement of a manufacturing process to ensure the quality of product and the consistency of manufacturer. We also need product-related data and testing plans that are adequate to support the manufacturing process and an appropriate facility. We do need non-clinical data to characterize the immune response and to evaluate and assess the safety preclinically. And of course, we need human clinical data that are adequate to support the proposed indication and use of the product. The product needs to be made in a facility that is in compliance with current good manufacturing practices. And we usually want a post-licensure pharmacovigilance plan so that the vaccine post-approval can be further assessed for efficacy and safety. This slide provides an overview about the vaccine development process, and I don't want to go into details here, just to mention that usually the development of the manufacturing process for a vaccine parallels clinical development. And clinical development, which is displayed in the lower half of this cartoon here, is usually divided into discrete steps, phase 1 and phase 2 and phase 3 clinical studies. Phase 1 studies present the first-in-human clinical trials to look at safety and immunogenicity in a small number of subjects. And then the clinical development proceeds to phase 2 studies, also evaluating safety and immunogenicity. These studies usually include a couple of hundred subjects and serve the purpose to look at the dose and the final formulation and sometimes preliminary effectiveness of the vaccine. And then finally, phase 3 trials, which are large pivotal studies to assess the safety and the efficacy of the product. If data are adequate, the vaccine manufacturer then will submit a document to us that is called a BLA, or Biologics License Application. And we will assess the data and the information contained in that document. And if data are adequate, then we will license the product. Once the product is licensed, there may be phase 4 assessments to further evaluate the safety and even the effectiveness of the product, perhaps in certain subpopulations, etc. So looking at this through the lens of COVID-19 vaccine development, considering the current pandemic, it's of course critical to have a safe and effective vaccine available to be administered to people to protect against COVID disease as soon as possible. And we're looking at very compressed clinical development timelines. So rather than doing the clinical development into the discrete phases that I just described, phase 1, 2, and 3, manufacturers do make use of adaptive and or seamless clinical trial designs that allow more rapid progression through the typical phases of clinical development. Of course, the data required to assess the safety and efficacy are the same. Now, because of this compressed clinical development timelines, there may be a bit of a risk that the manufacturing process that usually parallels clinical development would lag behind clinical development and may not be completed by the time phase 3 trials are completed. And so some validation operations of this manufacturing process may need to be completed once the product is approved, but only and only if product quality is not compromised. And usually there is an agreement between the FDA and the vaccine manufacturers on the type of validation operations that may be completed after product approval. When we look at COVID-19 vaccine development, there are 3 critical considerations, as for all preventive vaccines: safety considerations, efficacy considerations, and manufacturing considerations. So a key safety concern for COVID-19 vaccines is the potential for vaccine-induced enhanced respiratory disease when recipients of the vaccines are subsequently exposed to the wild-type coronavirus. Enhanced respiratory disease associated with human coronaviruses, however, has thus far been only demonstrated in animal models of SARS-CoV and MERS-CoV, and has been characterized by a Th2 biased immune response, resulting in eosinophilic immune lung pathology. Again, this has not been observed so far with SARS-CoV-2, and it's a theoretical concern only, yet the potential risk for vaccine-induced enhancement of disease or other immune-mediated reactions will have to be evaluated as part of preclinical and clinical development of these COVID-19 vaccines. And of course, there is a need for an adequate safety database and safety evaluation because these vaccines will be widely deployed to healthy subjects. We have been informing vaccine manufacturers that the effectiveness of the vaccine needs to be demonstrated directly using a clinical disease endpoint, and that is prevention of COVID-19 disease or infection. And hand in hand goes the need for pre-specified statistical success criteria that will ensure success of effective vaccines only and rejection of ineffective vaccines. And I already talked about the need for a validated manufacturing process to ensure consistent product quality, as well as a need for facilities that are functioning under good manufacturing practices. These key concepts have been captured in an FDA guidance for industry document entitled Development and Licensure of Vaccines to Prevent COVID-19. And we think that this guidance document will help facilitating the timely development of safe and effective vaccines to prevent COVID-19. It does reflect advice the FDA has been providing over the past several months to companies and researchers and other stakeholders interested in the development of vaccines. It describes the agency's current recommendations regarding data needed to facilitate clinical development and licensure of vaccines to prevent COVID-19. Now, time, of course, prohibits me today to really talk about the details of the clinical data required to support licensure. And so I just wanted to make some high-level summary points. Regarding non-clinical data, that is the chemistry, manufacturing, and control information, as well as non-clinical data, the guidance describes that COVID-19 vaccines licensed by the FDA must be adequately characterized and manufactured in compliance with applicable standards to ensure their quality and consistency. Non-clinical data and studies in animal models are needed because they help identify potential vaccine-related safety risks, and they guide the selection of dose, the dosing regimen, and the route of administration to be used in clinical studies. So sponsors typically perform repeat dose toxicity studies to assess the preclinical safety of the product. They do characterize the immune response induced by the vaccine in animal models, and they conduct studies to address the potential for vaccine-associated enhanced respiratory disease. I've mentioned in the beginning of my presentation the early phase 1 and 2 clinical studies to assess the safety and the immunogenicity of the product. And then in order to demonstrate the safety and effectiveness to support licensure, sponsors also need to conduct phase 3 studies to evaluate, of course, the ability of the vaccine to protect humans from SARS-CoV-2 infection and or disease. And we have said that an acceptable primary efficacy endpoint for these studies is laboratory confirmed COVID-19 or laboratory confirmed SARS-CoV-2 infection. Phase 3 clinical studies to demonstrate vaccine efficacy will likely need to enroll many thousands of participants, including those with medical comorbidities. And for those products that are in advanced clinical trials, in phase 3 clinical trials, they have been enrolling at a minimum of 30,000 subjects. FDA also encourages enrolling diverse populations most affected by COVID-19 in all phases of vaccine clinical development, including racial and ethnic minorities, as well as, of course, the elderly individuals, pregnant women and women of childbearing potential, and pediatric populations. The general safety evaluation, including the size of the safety database needed, should not be different than for other preventive vaccines that are being developed and licensed. The subjects in these studies need to be followed up long enough to evaluate safety, the duration of the immune response, and the risk of disease enhancement as antibody titers wane. FDA anticipates, however, that adequately powered efficacy studies for COVID-19 vaccines will be of sufficient size to provide an acceptable safety database. Post-licensure safety evaluations, as I mentioned, is an important consideration. So in summary, COVID-19 vaccines will be approved based on data from adequate and well-controlled studies demonstrating the safety and effectiveness of the vaccine construct. They will be approved based on facility data demonstrating compliance with current good manufacturing practices, and they will be approved when manufacturing processes are established that can assure product quality and consistency. Of course, each vaccine candidate has its own consideration, and the type of data and the amount of data and the will certainly depend on the vaccine construct that is under consideration. And we feel that continued engagement with stakeholders such as vaccine manufacturers, clinical trial sponsors, as well as national and international partners are critical for successful clinical development and licensure of COVID-19 vaccine. And this concludes my brief overview on the considerations that govern clinical development and licensure of COVID-19 vaccines. Thank you very much.
Thank you very much, Dr. Gruber, for this very informative presentation. I also would like to thank my other two speakers, Dr. Katz and Dr. Kreil. We will now have a short Q&A session. So please, if you have any questions to any of the experts here, please type them in. We will address them. So please feel free to send as many questions as you would like to. Maybe just to start off, I have a couple. I would like to start off with Dr. Gruber. You have stated that the vaccine trials that are currently ongoing and that they have not shown a common adverse events pathology, which other SARS coronavirus trials have shown in the past, which was a lung pathology. We know that from the press that 2 trials were stopped because of some medical problems in vaccinated people. What were these complications, and are they related to vaccinations?
So, first of all, going back to what I reported on this theoretical risk of enhanced disease, again, that has been only observed in animal models previously when vaccine candidates for MERS and SARS were used to immunize the animal models, and then they were subsequently exposed with wild-type virus. This is a theoretical concern for SARS-CoV-2. It is being evaluated as part of the non-clinical development studies that are being conducted for SARS-CoV-2 vaccine candidates. And even there, we have not seen any evidence for enhanced disease. But we also look at it as part of clinical development. We're making sure that the immune response induced by the vaccines is characterized in clinical assays. And we also try to observe for the potential for enhanced disease in these phase 3 clinical studies. It is true that there were some ongoing phase 3 clinical studies paused. These studies are under investigation, new drug application, and I cannot really tell you anything about that because these are confidential. But we have been working through this, and these trials, and I think there is some publication that they were allowed to proceed. So that's all I can say to that.
Thank you. And I have a second question from the audience. Have any BLAs been submitted to date to you?
It's a very important question, very interesting question. And unfortunately, again, I am bound by some confidentiality rules that I cannot really speak to that. Suffice it to say, companies who are in advanced clinical trials have made statements to the press that they are evaluating their SARS-CoV-2 vaccines in these phase 3 clinical studies. And some of them are very transparent and even publish their clinical trials protocols. And as you can see from what is, presented publicly, these trials are very much underway. Okay,
thank you. I would like to ask a question from the audience to Dr. Katz. How long can a person be symptomatic? And are they making antibodies throughout all this time when they are symptomatic?
Well, so I think my last slide or next to last slide was about this thing that we're calling long COVID. And we now know we're out 8-9 months into a lot of cases, and there is a substantial minority of patients with a variety of persistent symptoms for that long. I've seen that during my career with a number of other serious infections. Some of the people with long COVID, in fact, weren't very seriously ill, but have persistent symptoms over many weeks and months. Very little data that I have seen, other than anecdotes, that really tells us anything about whether the presence of prolonged symptoms associates or correlates in any way with the immune response, whether cellular or humoral, that is T cells versus antibody levels. And where antibody levels are concerned, what test is being used and the specificity for what component of the virus would probably be important. So these are things, like a lot that we talk about with clinical COVID these days. It's one of those things where I'll ask you to get back to me in 6 months and I might know more.
That was actually my next question. This is a question for me personally. You say that you see that quite common with other infectious diseases, that people have these long-term symptoms.
Yes.
Yes, and is that something we expected? Because we simply haven't heard about it that much. But is it a common feature of the virus
Well, no, it's certainly common for people with serious infections that have been in ICUs. The recovery period can be very long, and trying to sort out where those symptoms come from can be very difficult. Everything from post-traumatic stress disorder to severe deconditioning to ongoing immune responses with abnormal cytokine levels and whatnot. It's probably different for everything. It is not surprising, would be the point that I would make. And certainly in the United States, there's been an ongoing, long and difficult conversation about chronic symptoms from Lyme disease, whether that represents persistent infection versus inflammatory or immune responses versus more psychosocial things. And it's probably a mix of all those things. And my expectation is that we'll find that long COVID is a heterogeneous population as well.
Thank you, Dr. Katz. I have another question from the audience. I'm not sure whether I should direct this to Dr. Gruber, the vaccine expert, or Dr. Katz, the infectious disease expert. So how do you view the NIH recommendation that the COVID vaccine efficacy should be 60% higher than the 50% benchmark given by the FDA?
That's for Dr. Gruber.
Well, first of all, the guidance document that we have issued in June states that the efficacy, the point estimate should be at least 50%. So, and then with the lower bound of, the confidence interval around that point estimate of at least 30%. So we have to look at both. And the reason why we said at least 50% was because we wanted to make sure that there is not a vaccine licensed or approved that has mediocre or less efficacy than that. So, but in any case, I mean, if there's a vaccine that is 60, 70, 80% effective, we would very much welcome that. So we look at the at least 50% point estimate as the, if you want, sort of the minimum that we would like to see, that is reached, so that we can make sure that the vaccine that is then given to lots of people will have an impact. Over.
Thank you. Thank you, Dr. Gruber. I have a question for Dr. Kral-Thomas. So you state in your presentation that zoonotic viruses are known to jump species into humans. And we kind of have seen this on the virus examples that you've shown. Do you expect that we will see this more to come in the future based on various things?
Well, quite frankly, judging from history, it would probably be irresponsible not to assume that there would be further examples of zoonotic viruses. Just 2 weeks ago, we had another example published in Nature where it turns out that a mouse species widely prevalent in many parts of Europe carries a relative to rubella virus. It has shown to be lethally infectious to certain mammals, not yet humans. But I think these events are just absolutely possible. And it's not a question of if, it's a question of when we're going to face the next zoonotic agent. I think the only thing that behooves this industry, and frankly, the microbiological community in general, well, is to be prepared.
Thank you, Dr. Kral. I have another question for you from the audience. If the virus was detected in a donation, would you then ask this donor to donate convalescent plasma? So I assume it relates to the seeping through of the virus into the plasma, but I think we kind of, yeah, so I leave it to you.
Well, to respond to that, I'd like to defer to a guidance document that the FDA has published very early on the safety of plasma derivatives, where they stated that infectivity of these respiratory viruses is not known to occur in blood or plasma. So the scenario that was portrayed in this question remains hypothetical. And obviously, if we were aware that there were infectious virus in a plasma unit, we would not use that unit. But that's not a concern that I have around this virus specifically. More importantly, though, and I did mention that in my presentation, a review of the data available of this coronaviruses, all of the ones that we see in circulation, clearly shows to us that the inactivation and removal methods that were embedded into the manufacturing processes over the last decades are very capable of surely dealing with that hypothetical scenario such that product safety will remain uncompromised.
Thank you, Dr. Kral. A personal question from me on antibody kinetics. I'm fascinated. Do Do you, because you talked about antibody titers, so we have seen from your presentation that the antibodies peak around 18 to 20 days after symptom onset. And obviously, with regard to production of hyperimmune plasma, we would want to get those high titer donations. So would this be the perfect time to donate plasma to receive antibody titers? Because also antibody titers have shown to decrease with time after infection.
Yeah, that's quite correct. It's a very important question because it's not only the basis for the production of a hyperimmune, but also for the provision of convalescent plasma of adequate potency. I think it is not unheard of in immunology that you will see an early peak of antibodies and then somewhat of a decline. But I think many of the studies that have portrayed that antibody decline as being very dramatic are overstating what happens. There is a decline, sure. It's statistically significant, if you will. I don't think it carries a lot of physiological relevance, though, because a decline of twofold, say, that is just one dilution step of the functional antibody assays we have available to measure them. So there is a decline, and yes, there will be a need to select adequate donations of sufficient potency with respect to the neutralizing antibodies. But I don't necessarily view that decline as something that is going to render impossible the provision of these potent products.
Thank you, Dr. Kreil. I have a question for Dr. Gruber about going back to the vaccine trials from the audience. If a vaccine trial is stopped because of an adverse event, is it possible that the adverse event occurs to a volunteer who has received the placebo? Or would the trial only be stopped if the adverse event was experienced by the volunteer who has received the vaccine candidate?
Yeah, thank you for that question. So these phase 3 clinical trials are blinded, and that means neither the investigator knows or the clinical trial sponsor knows or the FDA would know which if what trial participant got either placebo or vaccine. So if there is an adverse event that calls for a stoppage of the trial, then one would look in unblinded data. There's something that's called the Data Safety Monitoring Committee that would take a look and then would provide information. Was the adverse event seen in the placebo recipient, or was it observed in the vaccine recipient? And then investigation would be initiated to see, if the adverse event was in a vaccine recipient, if the event could be considered vaccine related or not. Many times they're not vaccine related because adverse events do occur in people, especially in these clinical studies where we have a high proportion of elderly subjects enrolled which have comorbidities. So adverse events are not unexpected. But to make a very long story short, a clinical trial would be paused if there's an adverse event, regardless if it's in the placebo recipient or vaccine recipient, because the trials are blinded and you do not know at first, where in what participant the adverse event occurred.
Thank you, Dr. Gruber, very clear. And the $1 million question that I just have to ask to you again, how soon can we expect a vaccine for COVID-19 in your opinion?
I personally hope as soon as we have the data that support the safety and the efficacy of the product, robust safety and efficacy data that come out of these phase 3 clinical studies. So, and then of course, if we have the data and we have assurance that the manufacturing process is in place by which the product can be made consistently, we would accept a biologic license application and we would work very, very fast to get this product approved. And I will tell you, I've been 25 years in the Office of Vaccines. And one thing I learned, and this is never make prediction as to when a specific vaccine will be approved.
Thank you, Dr. Gruber. I have a question for Dr. Katz, because you also, of course, as your role as a public health professional, when you see it from your perspective, what are the best indicators that show an impact of the SARS-CoV virus on a particular country or region? Is it the number of infected persons, the number of hospitalized patients, the rate of mortality? What would you say?
Well, certainly in my jurisdiction and across the United States, we use all of those. On a day-to-day basis, I think the most critical thing that we watch is hospital capacity. The worst thing that can happen is to run out of hospital capacity and have people then have bad outcomes related to that. But it appears to us, I've got data since our first cases in March, that cases, hospital capacity, and mortality are all pretty proportional to each other when looked at over reasonable periods of time. And certainly the reported caseload is our first indicator of where things are going, so that hospitalizations, lag, case reports, positive test results by 7 to 10 days, ICU admissions, use of mechanical ventilation, and death 2 to 3 or 4 weeks following those case reports. So we look at them all. We probably pay the most attention to case reports and hospitalization rates.
Thank you, Dr. Katz. Yes, another question from the audience. I'm not quite sure who to address it to, Dr. Kreil or maybe Dr. Katz. What is the status of immunization of CPP and hyperimmune globulin donors who were previously infected naturally?
Well, we've distributed several thousand doses of convalescent plasma from my center, qualified using a variety of antibody assays. And we're able to deliver in real time. So we don't have orders from our 100 and some hospitals that take 2 or 3 days to fill, but it's very tight because it's very popular. It's very popular, I think, based on a couple of things. One, the absence of other highly effective therapies, and secondly, misunderstanding and desperation, a little bit of desperation on the part of clinicians who are misunderstanding the data available. I think the data available are strongly suggestive that using convalescent plasma in critically ill people is probably not going to be terribly effective, because by the time they're critically ill, in most cases, the virus is gone. There's no more viral replication. So we're trying to push in our system to get people treated very, very early, both because it's likely to be more effective, but also to preserve the supply for the cohorts of patients where it's most likely to work. It's tight, but in the US, we're getting by.
One last question to Dr. Kreil, and so we have seen reports of a small number of persons being reinfected with the virus. So they have gone through the SARS-CoV virus infection, they have been reinfected with a different strain, as in we can test that. How do you see the significance of this, considering that it has been receiving a lot of media attention?
I guess I would like to offer 2 aspects on that question. The one is reinfections, and we have yes seen a few reported. But I think it's important to keep in mind that this is on the basis of north of 40 million people who had gone through apparent infection. So that reinfection, as I can see it, is a very, very rare event. And it may just be that the immune system of the persons affected is not fully intact, and therefore it may just represent a very small proportion of the population and should not, I believe, be generalized to say that reinfections will occur. The second thing is about that second strain of virus that apparently is out there, so that their reinfection might be possible. I have not seen the evidence to support that. To the contrary, there is very large studies to the tune of almost 20,000 genomes being analyzed. And if anything, it would appear that the virus is undergoing what is called a purifying evolution. So it's a very narrow evolution. And I have not seen the immunological evidence to say that they are not being cross-neutralized. So right now, the assumptions, I think, for convalescent plasma treatment, hyperimmune globulin treatment, and also vaccine development needs to be that we are facing one virus, and therefore any of these treatments have a good chance of being successful in the end.
Thank you, Dr. Kreil. With this, I would like to close the Q&A session. I would like to say thank you very much to my expert speakers who have really made the time to be with us today, and thank you to the audience. Bye-bye.